Hey, good morning, everyone. We're so excited that you're here. Today is Valentine's Day. Both of us are in our reds. Awesome that you're taking time. Those of you that are watching today, thank you for taking time out. And especially we want to thank Dr. Ruchi Sharma. So wanted to just again, welcome everyone. This is a series that we're doing. I believe we're on our fourth one now on potential treatments for choroideremia. So my name is Beth Foss, and I am the Director of Operations at the Foundation. And today we're having a discussion with Dr. Ruchi Sharma. And Ruchi, we met, I was trying to think of this with Eric Hartman, maybe 7 or 8 years ago, would you say?
I think it was 2018 or something.
It was 2018. It was during, we've seen you several times at the NIH Rare Disease Week, Rare Disease Day, had lunch together. And you were one of the original doctors too, and your team came for our SAB meeting.
Yeah.
And we had a great dinner sitting across from each other. Do you remember?
I know, I do.
It's great. And we're really very, very fortunate and grateful that you're continuing to work on IRDs, choroideremia, and just grateful to your team. So today you're going to be sharing some information with us about some really technical things that I have written down I could reference, but we certainly want just to give it to you. It's very interesting. I think your presentation today has garnered the most amount of folks that want to watch it. So it's super exciting. And as usual, everyone, I'm going to be taking the questions at the end or periodically if Dr. Sharma stops. So I'll be running the chat feature or the Q&A. So go ahead and send those questions in. We'll take time at the end, certainly, to get to your questions. And we always want to make sure we thank our partial sponsor, which is 4D MT. And I think that's it. We'll go ahead and get started. All right. Thanks.
Thanks, Beth. Thanks. I'm very excited to be sharing our work, especially on the Valentine's Day. And like Beth said, I met the family members of this foundation in 2018. And since then, I have been eagerly wanting to contribute more and work with these people. And I'm very excited to share our work and our approach that we can translate to for potential therapeutics of choroideremia. So like Beth said, my name is Ruchi Sharma. I am a staff scientist at NEI. I work with Kapil Bharti's group. And before I get into the details of my talk, let me just give a little brief overview what our lab does. So we primarily work on stem cells. And we derive these stem cells from blood or fibroblasts, and this blood and fibroblast is coming from patient or healthy donors. And what we do when we have these iPS cells that are called induced pluripotent stem cells, and I will explain in detail later also what these stem cells are and what we do and how we derive. But the overview is that we we make iPS cells from the patient and donor or healthy donors. We differentiate this iPS cells into this tissue that we primarily work on in our lab. And we use this patch either for cell-based therapies or for modeling disease. We model several retinal degeneration diseases like monogenic Stargardt, or polygenic age-related macular degeneration. So we do this modeling either just RP alone and some other tissues. And then our goal is to find the drug targets that we can take back to the patient and we can have potential therapeutics in terms of small molecules. So the idea and the lab focus is getting the potential treatment for retinal degeneration either through the cell-based therapies or through the small molecules. So okay, so let me just give a little brief overview of the layer I'm going to talk about and why it is important. So behind at the back of the eye, there are different retinal layers, and they are called so there are 7 layers, and they are like sandwiched on top of each other. the layer that we work on is retinal pigment epithelium. It is situated between photoreceptors that are the photosensitive cells of the eye that enables you to see in dark or in the light. And beneath RP there is a choroid. So this RP helps in the maintenance of photoreceptors and choroid, so that this homeostatic unit or outer retinal layers can function well and person can have a normal vision. If I show you in a little zoom out view and in the schematic, this is where the tissue is. You have photoreceptors. These photoreceptors are broader cones. You have a retinal pigment epithelium. That's the layer of tissue that we work on, and we make it from iPS cells, and the choroid, that's the blood vessel that is beneath the RP, and that nourishes this layer. And in choroidemia or in other retinal degeneration, either there is a degeneration of this layer or this layer or this layer, and eventually causing the vision loss. So there are different types of vision loss, and like there is macular degeneration that affects the center vision. So basically in the eye there is a central part that is called macula that is important for center vision, that causes that so in this disease you have the loss of center vision. In other retinal degeneration you have the loss of peripheral vision. But regardless, the bottom line is that these different retinal degenerations, be it's genetic retinal degenerations like choroidemia or Stargardt or retinitis pigmentosa or Best disease, or be it a polygenic macular degeneration like age-related macular degeneration, we need to have treatment for these disease. And there are different ways that people approach to have a treatment for these diseases, and particularly for the genetic disorders. There could be a gene therapy where what you do, where you really just, so in what happens in case of genetic disorder, there is a gene that causes and the mutation or the dysfunction or non-function of that gene causes the degeneration of that or causes a vision loss in that particular individual. So what you can do, you can literally transfect that gene and in the subretinal space, and then that can potentially replace or have the functional gene, and you can that can be a potential therapy for choroidemia. There are different companies and universities that are working in this space for choroidemia. But in our lab, our main focus, like I said, we work on stem cells. We get the blood samples from patients from different diseases like Stargardt or AMD or choroidemia or late onset retinal degeneration, or even in the healthy subjects. We get the blood cells, we isolate the particular cell type from that, and then we make the iPS cell, and then we make RP. So the idea what we have and the principle that we are working on to find a treatment for any retinal degeneration is to transplant a young or healthy our iPS-RP derived patch, and so that this patch can help in nourishing the photoreceptors on the apical side and choroid on the basal side. And we believe that once we replace or once we provide this tissue or the healthy patch in the patient's eye, that will restore or at least stop further vision loss in that particular individual. So this is the approach that we are currently using for age-related macular degeneration, and I'm going to show the data how we make this cell and where do we stand, and what is the and which stage of the disease this stem cell based therapies or the replacement therapies can actually work. So let me tell you about the stem cell. So there are different types of stem cells. The stem cell that we work on is iPS stem cell, this or induced pluripotent stem cell. So this induced pluripotent stem cell, the reason it's called induced because it is induced from a somatic cell. So for example, like I said, you can make a stem cell or iPS cell from a fibroblast, from a blood cell, from an epithelial cell, from your hair cell, from you name it, and you can make iPS cell from any somatic cell type. And the stem cell has a cell is called the stem cell when this stem cell when this cell has the potential to replicate, potential to differentiate into any other cell type. Like I said, in our case, we are using these iPS cells to make retinal pigment epithelium. People, as you might aware, there are different labs that use this stem cells to make photoreceptor cells. They make endothelial cells. They make kidney. They make ear. You name it, and you can make this that particular cell type of a body from the stem cell. So there are different kind of stem cells. Like I said, there are embryonic stem cells. As the name suggests, these stem cells are derived from embryo. So when so what happens when there is an embryo and the stage is particularly called blastocyst, you isolate the part of this embryo and then you culture them in a dish. And when you culture these cells that are able to differentiate into a cell type, and they can replicate in a dish. So, but what happened, as the name implies, there are various ethical issues that comes up with embryonic stem cells. So then in 2007 and 2008, there was a paper from Shinya Yamanaka's group and the lab in Boston. They derived iPS cells from somatic cells, and this iPS cells actually took over and overcome all the issues that you could think with the embryonic stem cells. So these iPS cells literally replace the embryonic stem cells. And these iPS, another thing is that you can generate these iPS cells from any patient. So then that way you can actually recapitulating the patient genetics also in a dish. So these stem cells, they behave very similar to each other. They are pluripotent stem cells, means they can make any other cell type. But we do have a tissue-specific stem cells. They are multipotent. For example, the blood stem cells, they can make the specific type of cell types. They cannot make epithelial cells or RP or photoreceptor cells. So in case in stem cell therapies and in case of disease modeling, this second number has taken a has overtaken the embryonic stem cells and other another type of stem cell. And this is the stem cell that our lab work on, and this is the cells that we use to generate RPs. Like I said, these iPS cells you can use to make any type of the any cell of the body. You can make endoderm, lungs, you can make RP, you can make choroid, you can make retina. So once we have these cells, and in my lab overview, I was suggesting that how we are approaching to have a potential treatment for the for any particular retinal degeneration. So in case of in our case, what we are doing, we're getting some cells, we make iPS cells, and we either make RPE, just RP alone. We either make this RP and a choroid, like I was showing you in my introduction. There's a RP and there's a choroid, and you can actually we can actually 3D bioprint this patch, and you can and imagine that in the extreme disease condition where the where the patient has lost their RP and their choroid, and that in the extreme stages or late stage retinal degeneration, this patch would be more useful or more workable than RP. With our collaborators like David Cam and others, we have been working with photoreceptors part 2. So at the end, the dream is that we have RP on scaffold, we have choroid beneath, and we have photoreceptor. So in a way, we can have the complete sandwich of outer blood retina or outer retina, and we can transplant that. We are not there yet, but we have at least we have achieved this stage, and we are in process of working on this stage. And this is the work that we are doing with our collaborators. what I'm trying to do in this my talk is that I want to share our experience or our journey that we took in last 10 years for getting a food of FDA approval for our stem cell therapy. So the way this is an overview of any cell therapy that you want to take it from the bench to the bedside. By bench means like a lab. So what you do, you basically develop your protocols. You do a proof of concept studies in vitro. You do those concept studies in animals because you want to show that the product you're making is going to be safe when you're going to transplant in patient. But before that, you use animal models. Like in our case, we use pigs and rats. And then you do IND enabling studies. That FDA requires you to do that, and that takes almost these many years. And then once your package is ready, you have shown that your product is not toxic. You have shown that your product is indeed is efficient enough to in your animal model, whatever animal model you choose, and then you submit your documents to the FDA, and if FDA approves your IND, you can start phase 1, phase 2, and that's where we are right now. So we did all this part. We submitted our IND to FDA in 2019. We got approval right before the pandemic, and last year we had first patient transplanted for AMD, and I'm going to show you the data for that too. But the bottom line is that this is a long process, and before any therapy reached to the patient, there are a lot of work that goes into that. You have to have your product protocols in place. You have to have your proof of concept, your data in animals done, your toxicology and your efficacy studies done. So I mentioned the name IND. So IND is what does it mean, and why, what are the different parts that constitute IND package? So IND is an investigational new drug. So when you are working on new therapy and you want to get for an FDA approval, there are different aspects specifically for the stem cell based therapy that you need to cover. You want to show that the manufacturing can be done. You have to do the clinical grade manufacturing process. You have to because for the patient and for the clinical studies, everything is done in the clean room, or you call the GMP manufacturing, and then you validate this clean product manufactured in the clean room in your through in vitro or in your animal models. And like I said, animal models is preclinical models. That's called preclinical data. That means before it goes into clinical, that is a preclinical data. You want to make sure that there is no toxicity or side effect of your product that you have generated from iPS. You want to make sure that the efficacy of the product is you have to show the efficacy of your product in your animal models, and then of course the compatibility of the tool, the surgery tools that you're going to use in your procedure. And this is the more of a clinical side that works with you, where you have a clinical protocol set up and the consent forms ready, which you're going to share with the patients, and they know what they're getting into for the clinical trials and what they agree to and what they don't agree not to. So when you work with iPS cells, or I would say or stem cells, and particularly when you want to make a tissue from a stem cell and you want to use that tissue for your cell therapy, you want to have a protocol that generates pure and mature iPS, that particular cell. In our case, I said this is RP cell. So what we did in our proof of concept studies, we optimized this protocol to generate RP cells starting from the iPS cells, and we wanted to make sure that the cells we are getting at the end of this protocol, they are mature in terms of their molecular signature, in terms of their functionality, in terms of their structure. So this is the timeline, or this is our protocol that we use to make RPs. Starting from the iPS, I'm not counting the time that it takes to make iPS cells from a blood cell, but from iPS to RP, it takes almost 10 weeks. And once we have these RPs, validate them in with using bunch of assays that lab has established. But in the bottom panel, you can see this is a well of a 6-well plate, and you can see this is the homogeneous RP cells that are pigmented, and this is this plot here just shows you that the cells that we are generating in vitro, they are more than 95 or 98% pure. So another thing is that when you are working with IPS product, or you are working with a protocol that generates the product that you want to take it to the patient, you have to show the reproducibility of that protocol. So we have shown, and we have done more than 100 lines of differentiation through this protocol. Here, some of the disease and the healthy individuals are listed here. So once you optimize this protocol, and you need to take it to the clean room, like I said, where you actually going to manufacture the clinical product, you streamline every process. Since our protocol is long enough, and we want to make sure that the product we are generating from this iPS cells, they are that is pure, and it doesn't have any sterility issues, by means there is no mycoplasma or any other contamination. So we have in process quality control assays at different stages, where what we do, we check the bank of that we generate from during the process of manufacturing for sterility. We want to make sure that this iPS cells we are generating, they don't have any abnormalities in terms of any mutation. We want to make sure that the cells that we are making from the patient, they actually belong to the patient. So that is called identity. And then when we set up this stem cell bank to the differentiation, since we have separate different go and no go steps, where we check for the efficiency of the protocol and we want to check this the purity of the product that we are generating. So we have several flow based assays that we use, and we have cutoffs that we say, okay, well, this round that we made RP doesn't pass this because it did not pass the threshold that we set up. At the end, before this product is released for patient or any other assays, we do validation. We want to make sure that RP is indeed RP in terms of its functionality, in terms of its maturity, and make sure that there is no that the sterility is not compromised. So then, okay, so our approach is that when we differentiate these iPS cells to RP, and when we want to take this patch in the patient eye, or when we want to implant this patch in the patient eye, before that, we culture our cells on this biodegradable scaffold. And this biodegradable scaffold is made of a PLGA, polylactic co-glycolic acid. And the reason we chose this PLGA scaffold, couple of reasons. First, when you want to transplant something, you want to have some physical strength, and we did not want to have a plastic. We did not want to have cells this culture in the plastic and implant plastic in patient's eye. So we chose biodegradable scaffold. Other thing, we did not want to leave anything in the patient's eye other than the cells that we are implanting. So we chose a biodegradable scaffold. So once we differentiate the cells, we have our RP cells. We seed these cells on the scaffolds, PLGA scaffolds, and we culture these cells for 5 weeks. So this is a image that you get once after 5 weeks of culture on scaffold. This is RP, the hexagons. You see RP cells, and these are stained for Z01 and beta catenin that marks the membrane of these cells, and this blue color is for the nuclei of these cells. Each cell has a nuclei. So this is another way of representing how the cells or iPS-RP cells look on the scaffold is this pseudo-colored SCM image. So this is a scaffold, the blue color. The brown is the RP cells, and this is the nucleus. And the this green color over here that you see is the apical processes of the RP cells. So these are this is one of the important structural feature of RP cells, because RP interacts with photoreceptors to do certain functions that are important for the maintenance of the photoreceptor cells. So this is just one way of showing that cells that we made and the cells that are grown in the scaffold, they have nice apical processes and they have a nice monolayer. So I did not cover a lot of assays that we do in vitro to confirm the functionality and maturity of these cells, but I'm happy to answer any questions anyone have about those assays that we use, and we have published those assays in detail in our papers also. But like I said before, before you do your IND enabling studies or you get approval, you need to show proof of concept or preclinical efficacy studies. In that case, you can have different animal models. We chose rat, and we had a pig animal model that I'll show you in a bit. But what we did, we transplanted a 0.5 millimeter diameter piece of this iPS-RP patch on scaffold in immunocompromised rat. Over here, you can see this is image, I believe, after 10 weeks of the transplantation. But the point what I want to make here is that this layer, which is stained for stem 121, which stains human cell, the red color you see here, is it represents the iPS-RP patch that we made in the lab. And if you can appreciate, you can see how well this patch is integrated in rat RP in the rat RP layer, and this is a rat choroid. So this is the one way that we showed that the patch that we implanted in rat eye, it indeed integrates with the rat RP. The other model that we use was is pig model. But before that, let me show you some data that we our study design that we used to confirm that the cell that we generate in our lab, or these iPS-RP cells, they don't have any toxicity or tumorigenicity or effects when they were injected or transplanted in the rats. So we did this study in rats. We did this in collaboration with Covance in Madison. We had almost 450 rats that were transplanted, and we had different short-term and long-term follow-ups. And we looked for the toxicity. We looked for if this dose that we transplanted in the rat, if it caused any kind of toxicity in that space. By that I mean there's an immune response or not, and then if when we transplanted this patch in eye, we didn't want to see that this patch or the cells migrate to other parts of the body, or in liver or kidney, or crosses other layers of the eye. So we did the biodistribution study, and our last study was the tumorigenicity study. That means that we didn't want to have any tumors develop because of our iPS-RP patch that we are transplanting. We did not see any side effect in any of the 450 rats that were transplanted, and this was a big study, and it took the last time point was 9 months, and even after the 9 months, we did not see any side effects or any kind of biodistribution or the cells migrating to any cell type or any other part of the body when we were transplanting, when we transplanted this patch in rat eye. Then let me move to the our porcine model.
Hey,
So
Dr.
this
Sharma, can I ask
Sure.
you, I have two questions. Now we can wait to the end, or I can ask them now. Which would you prefer?
I can answer now.
Okay, this is just a quick question. Does the existing retinal sandwich have to be removed before the newly grown retinal sandwich is inserted? In short, do you have to do a retinal demolition before the new healthy retinal component is inserted?
So I assume this question is for the animal studies that animal data that I am showing. So in our pig model, we did laser injury. In the rat model, we did not demolish the RP cells existing in the rat. So in a way, this is true that the human RP cell that we transplanted in the rat, they were actually sitting on the pig RP cells. Oh, sorry, on the rat RP cells. But in our porcine model, that I'll show the data, we actually laser ablated the RP, and we tried to recreate the disease condition, and we tried to transplant it there. So what I will say, the rat studies that we did is kind of a more of a safety model that we wanted to see that if it integrates or if it causes any tumors. But the efficacy model or efficacy studies where we want to see if actually the implant we are transplanting, it will do something, that is the pig model. I hope I answered the question.
You know what, if you didn't, because I did not understand a lot of that, but so if you didn't, I'm sure she'll right, she'll ask a follow-up question. And here's a general question again that maybe you want to save to the end, but just think about it, or you can answer it now, certainly. I believe this treatment is not available for my 15-year-old son. His CHM is very aggressive. My question is, can stem cells revive the retinal cells once very little vision is left?
So, I mean, so I think yes. So the answer is that if you are going for the late stage retinal degenerations where you barely have any other layer, for example, so then I think that in that case, you will have to have RP, you will have to have a choroid, you will have to have a photoreceptor. In cases where like the AMD, the approach or the data that I've been showing is like, we are transplanting this RP patch where we know that there is some photoreceptor, they are dying, they're not completely dead. You know, so I guess what I'm trying to tell you is that depends how many layers or how many which sandwich do you need depending upon which disease stage you are targeting the patient at.
I think that makes sense. I think that answers the question. Yeah, thank you. Go ahead. Keep going. You're doing great. Thank you.
Thank you. No worries. Okay, so I was talking, so this is, so the rat data that I showed before, it was primarily more of for us to have to show that the cells that we implant, they are safe enough. Because the rat model did not show any, did not recap or did not have any retinal degeneration. So I don't think we can conclude anything about the efficacy of the product, of our product. For to do that, to do efficacy studies, by efficacy, I mean if the transplant is able to revive the visual functions in the eye, we developed this pig model. And what we did in the pig model, we as you can see in this schematic, we laser ablate or we laser injured the RP cells in the pig eye by using laser of particular wavelength, which is 532 nanometer. And after 48 hours of treatment of this laser, we use this pig's eye for implanting the patch. So if you see on this on the left of the slides, you have this infrared image of the pig eye, where you see these different windows of patches, right? And the percentage, 1%, 1.5%, 2%, it tells you the intensity of the laser that we use to ablate the RPs. I am making it I'm making it a little simple, but it's not actually the intensity. But just think about like this: by 1%, what we saw, we were able to kill the RP cells, but it was a bit too gentle. And if you leave, the RP cells will recover back. By 1.5%, we were able to destroy RP or kill RP. By 2%, we were able to kill photoreceptor layer and RP layer. So but what I'm trying to say here is that what we try to do with this laser injury model, and this paper is under review. Hopefully, you guys all will be able to read soon. We were trying to recap the different stages of retinal degeneration. And today I'll just show you the data about 1.5%, because that's the one that we have been using for our iPSC-RPE batch. Then there are different assays and techniques that surgeon and the preclinical team does to assess the health of the retinal layers. There is this optical coherence tomography. There is other to see the choroidal blood vessels. They do this fluorescent in angiography for seeing the cones. They are doing the adaptive optics. There's different techniques that we use. I'm not going to talk in detail about these because I think they're too technical to discuss, and even beyond my knowledge coverage area to talk little in detail about these. Anyways, so for choroidemia, what we are currently doing, or what we have done, we wanted to develop a pig model that can recap the degeneration that you see in choroidemia patients. And we got this grant in collaboration with David Cam, where our goal was to transplant this iPSC-RPE patch in also we had two goals. At first, we wanted to just transplant the iPSC-RPE patch alone and see how do you see what does it do to the choroid beneath. Or we had another approach that we did with David Cam, and this work is still ongoing, where we wanted to transplant the dual. We wanted to transplant the photoreceptors and the RPE, and we wanted to see, does it help maintaining the choroid, or does it or which one is better in efficiency? We have a lot of data. On iPSC-RPE, but currently we are still working on the dual, so I would not be able to show you much of that data. But the idea is, the point is that I'm trying to make is that we are using different approaches and different ways to mimic the disease and then do the transplantation. So this is once we do this laser injury in pig eye, we wait for 48 hours, and we do the transplant implantation in after 48 hours in this laser injured pig eye. So this is pig OCT, or image of after laser damage, 48 hours, and this is, you see that this it affects the retinal layers. It has certain effects on the RPE also. Let me show you some recent data on this model and on this part of the study, which is currently under the revision, and we hopefully that paper should come out soon. So this seems very busy slide, but I'll just talk very simple language, and I'll just try to explain what we're trying to do here. So like I said, we use different laser power to affect the RP layer or the choroid and so on. In this data, we have we use 1.5%, and if you see over here, this is a OCT scan. This yellow thing, this yellow color, it shows you the choroid. So what we try to see, we try to analyze the effect of 1.5% laser injury on choroid. We wanted to see what happens there. So in here, if you see, you have laser injured area, and this is a healthy area, right? So because we laser injured the area, what we see, so when you see this yellow line, this signifies the presence of choriocapillaries. When you see this artifacts or these shadows, it signifies that this area does not have choriocapillaries. So what we did here, you see that with the laser, this is the laser area where we laser injured patient, pig eye, and this is the healthy area. So you see choroid here. When you laser injure the pig eye, you don't see the choroid here. This is pretty visible here. So what we did, we kind of follow up these pigs at 2 weeks, 6 weeks, and 12 weeks, and we noticed that this injury that we did with 1.5% of laser power, it does not the cell does not come back, and you still have the loss of choriocapillaries in this area. And then what we wanted to see, so this is a side view of OCT and this is a top down view over here. That's what you see. So this is a laser injury here area, and you see that with the time you do have some from the edges the cells grow, but then still there is loss of choriocapillaries. That's why you see a lot of these blood vessels, because choriocapillaries is gone. So then we wanted to see if we transplant our patch, does it help does it help recovering the choriocapillaries? Before I go there, this is the tool that we use for transplanting. The surgeons developed this tool. This is the tip of the tool that you can actually see the scaffold is loaded in the right direction. And then this tool is controlled by foot pedal, and you can see how surgeons control this loaded tool with the instrument that is controlled via foot pedal. And again, this is the tip of the tool where you can see the scaffold is inside. And the one thing is that the curvature of the tool is kept in a way that when the surgeons are going it matches the curvature of the eye, so that the transplantation or the surgery steps are more feasible. This is a little video of the transplantation, and I'm not going to basically what surgeons do, they make a cut in the sclera. This after they do and then you see there is a cut in the retina, and this is the RP alone patch. You see they're gonna release through the tool that I just showed in here. The tip of the tool is transparent so that you can actually see that the implant is transplanted or is released, and then they seal the retina, and then they from that point onward the follow-up of PIC starts after that. We do a long follow-ups and short follow-ups. So like I said in my previous slide, that when we did the laser injury of the pig eye, we lost the choriocapillaries, and that's why you see the blood, you see the choroidal vessels here. But after the implant that you see in here, that we did after 48 hours of the laser injury, what we noticed is that this is a patch, and you could see that this patch and this laser area, you can totally see the differences. You don't see the choriocapillary, you don't see the you don't see the blood vessels down, because we believe that this RP patch has been able to rescue the choriocapillaries in the transplanted eye as compared to the laser area. There is a lot of data and a lot of information I talk, but this paper is under revision, and hopefully all the details what I'm talking here, they will be you guys will be able to access soon. This is another way that we checked that indeed the transplantation of this patch had an effect on the retinal layers. So this is the outer nuclear layer. You see that the thickness of this layer was higher than the laser injured area. We did this is another view that you can that I really like, that really clearly shows you that when you have iPSC-RPE patch, if you see this area with the laser area, you totally see how these retinal different layers are much healthier than this layer, or even the empty scaffolds. This is I believe this is a 10 weeks follow-up, and this just the other thing I want to tell you, that you see that the retinal layers or outer nuclear layers are completely gone in laser area. But when you transplant it, you can see that this one is very similar to the untreated layers. So untreated area of the pig eye. So I think what I'm trying to tell you, and I think what we have seen is that this implant or this PLJRP that we transplant in pig eye, it indeed rescues the retinal layers on top of the implanted area. I did not show all of the data that all of the functional assays that we did with the pig's eye, because I think I think it's going to be too technical. But we do multiple assays, functional assays, and we do see that the retina above the implant or the RP patch is functioning similar to the untreated area or the unlasered area. So we all this work, it led us to the submission of our IND, and we got the approval from FDA in 2019, and we had our first patient transplanted in last August. And this is just basically like I said, we check for them, we make sure that the product is pure, there is no stem cell there, they are expressing high RP markers, and their functionality is high, and there is no compromise on sterility or mycoplasma, and there is no endotoxin. So once this all tests are done, then product is released to the surgery suite, and where we had surgeons from Wilmer Eye Institute and our own surgeons. They transplanted this first patient in late August, and so far what we know is that patient is doing well, and we hope to transplant the second patient in hopefully in late summer, or yeah. And then we are, so this trial has 12 patients, and we hope that we'll be able to do 2 or 3 patients this year. I did not realize that I might be going over time. Beth, I would like to ask you, so I do have some slides. I can stop here, or I can talk about that. Because I just take the time, and it's already 10:50, and I believe I might take like 5 to 10 minutes more to finish the slides I have, but you let me know.
I think you can take about 5-6 more minutes, because
Okay.
we have already about 6 or 7 questions in the queue, and I know more will come in. So we like to end about 11:15 at the latest. So we'll plan accordingly.
Okay, thanks.
Mhm.
Yeah. So I think Beth asked me a question about like which stage does the patches alone will work, and which stage will we need to have we need to have the sandwich of RP choroid or photoreceptor RP. So I think like I said, it depends on which stage of the disease we are targeting. For example, gene therapy, for any gene therapy, you need to have at least a healthy cells that can survive and that can function. For this, I believe the replacement therapies come little later, because you actually, as the name suggests, it's a replacement. You want to replace the dying or the dead or the missing cells, and implant the healthy cells there. So in the beginning, I said that we so far I showed you the data or our lab efforts or work on RP alone. But in lab, we do also print RP and choroid. So for that, you need different type of cell types, and we published this paper recently in Nature Methods, and you all are welcome to check this paper, because it's so the idea is that we want to generate RP, and we want to generate a choroid beneath that. And for choroid, there are different cell types you need. You need endothelial cells, you need pericytes, you need fibroblast cells. So what we did, we used this approach of 3D bioprinting. We used this 3D bioprinter, and then what we did, I would like to, oh, sorry. I don't know what I did. I clicked on something stupid. Yeah, wait, sorry, let me go back. So what I was trying to tell you that we use this 3D bioprinting approach, and we literally printed the cells in a bioink, and we call this bioink because this ink has the cells, pericytes, fibroblasts, and hydrogel. So what we did, we and what like and just to remind you all what we are trying to do here, we want to create this tissue, this sandwich, RP and choroid. So what we did, we used the same approach that I showed you for our RP patch. We used the scaffold, and on one side of scaffold, we cultured RP cells. And on another side of the scaffold, we culture these cells. So this is the bioink that we printed in this format, and this is the vessel that these cells tend to make in the empty space. And after a week, we flip the scaffold and we culture RP cells on it, and we culture this sandwich or this out of blood retina barrier for 5 weeks. And then we did a lot of assays to make sure that these cells have the Brooks-like membrane. We check for the functionality of this tissue. And the bottom line is that you can see here, you can see how these vessels are in this tissue. So this is the tissue that we printed, and these are the vessels these are stained for different markers. But you can see how this tissue after 5 weeks imprinted, cultured, and you have RP on top. You can see how there are vessels generated. So the another video that will show you in more detail. So this is choroid at the back, and this is RP on the top, and this is the colored images of this tissue. You have choroid here, and these are the density images. So you have the bottom choroid, on top you have RP. So what we just like last example I'll show before I end is that with this tissue that we created, we try to see if we can recap or we can use this tissue to model any kind of retinal degeneration. Here we use this model to we use this tissue to model wet AMD. In case of wet AMD, as you guys might know, what happens that the blood vessels, they tend to poke RP cells, and in extreme cases or the late stages of wet AMD, you see the blood between the subretinal space also. So what we were trying to do here, we tried to mimic the situation or condition that happened in wet AMD. And when we did that, you will see, or you will appreciate that these circles that you see here and the arrows, these represent the blood vessel. You see how these blood vessels get much closer to the RP cells when you compare this to the healthy condition or the untreated condition. So the idea was just to see if we can model, we can use this tissue to model the different retinal degeneration. And like we all know, wet AMD has this anti-VEGF therapy, and when we treated this tissue with anti-VEGF antibodies, we saw that you can actually regress the protrusion of these vessels to the RP surface. So so in the end, I would like to thank all the people that I have been fortunate to work with. This is my PI, Kapil. I'm sure most of you guys would know. This is a picture we took after the first patient was transplanted, and we were waiting for the surgeons here to just welcome them all. This is our preclinical team. This is our project manager, and this is our surgeons. And I feel I'm very fortunate that I get to work with all these people, and I'm very thankful to you all that you joined me and listen patiently the work I shared with you all. And I'm happy to take any questions you might have.
That was amazing. I was just texting Kathy Wagner, our executive director, and I said, this is mind blowing. This kind of work that you're doing, it's astounding, especially live cells in printing. We do have a lot of questions coming in, so if we can get started, is that okay?
Sure. I can,
okay.
Stop sharing.
We're going to start off with the chat feature. Is CHM a disease primarily of the choroid, the RPE, and or the ONL layer?
I wish I had a straight answer, but I think so. What I know from my what I know, I think choroidemia does affect RP and it affects the choroid vessels too. So I would say it does affect. At the end, it does end up. At the end, this disease does end up affecting all the retinal layers. But I think it starts with RP. But again, this is my bias view because I work with RP. But yeah.
Okay. Lisa is asking, is the main focus of the stem cell therapy to restore vision, or are we mainly hoping that it will preserve the vision? That's a great question.
Yeah, I think it's more for the preserving vision.
Okay.
Yeah.
Eric Hartman says hello. So thank you for this great talk. When creating RPE cells from iPSCs over the 10-week period, are you doing all of the cell maintenance and cell differentiation manually, or have you developed robots to handle these exacting and very repetitive tasks? If so, are you also doing so for the differentiation of rods and cones for the placement in the scaffolds?
So this is there's so many things I did not touch, but this is very important point. Specifically, when you work for autologous cell-based therapy, by that I mean you want to create RP for a particular patient. Let's say, for example, I'm a patient, you are a patient, but then I'm going to get iPSC for you, I'm going to get iPSC for myself, and so it is very repetitive, and it needs a lot of technical expertise also. For now, whatever we are doing in the lab and in the clinic is all manually, but we are working with a company, and this company they are trying to automate the IPS part. Since it's a company in Boston, Celino, they are trying to automate the IPS part, and we are working in collaboration with them, and we are hoping that we will be able to automate the RP part too. What I know for the photoreceptors, I know David Gam's lab, they are generating these organoids or precursors or whatever you want to call them, photoreceptors in bioreactors. So I think they're already doing it. For RP, we are, because it's monolayer culture, it's not like a suspension culture. So you need, so we are working with this company, and we're hoping soon, as soon means years, we will have that. Yeah, that's a good point.
Good, thank you. Here, Marie is asking, what immunosuppression regimen did you use in the pigs?
I am not a, yeah, I can, so this thing is published in our paper, but the prednisone, I wish I'm not surgeon or clinician, but we do, and I can definitely tell you in detail what are the immunosuppression we use. The paper we published in 2019, it has it, and the paper that I was talking about is going to come out in JCI Inside, that will also have it. But again, if you want to, I don't want to just say random things because I know they do. It's better that I give you a right answer, but I need to check that, and I'm happy to email you back if you want me to.
Maybe you can share that with us, and we can put it on our web. Yeah, we could do that. How soon do you think the paper will be shared?
How soon? Sorry, I missed it. How soon?
You said that there's going to be a paper release fairly soon.
Yeah. Hopefully we should be able to submit in a month. You're talking about the paper that is in revision, right?
I'm guessing so. Yes.
Yeah. And but for this for these drugs that we are doing for compromising the immune system in pigs, I can I can send the information to you, and then you can share it with others.
Okay.
Yeah.
Katie is asking a question. Have you seen any inflammatory responses, immune responses in your animal models or in the trials? Do you have any assays that measure this?
So in animal models, yes, we do see that. The other reason we do see that is because the what we are doing, and the reason we are also immunocompromising the pigs is because we are transplanting human cells in pig. If had we have done pig cells into pig, you would not expect that. And our rationale is that because the approach that we are doing for the trial at the NEI for macular degeneration is autologous. So we are making patient cells and putting his own cells. So we have transplanted one patient, and what I know is patient is doing well, but other than that, I don't have any other information. But so far, the reason we do the we do this drug treatment in the pigs is because we are implanting human cells. We are currently doing pig cells in pigs, and we are doing it without any compromising the pig immune system. So we are hoping that will give us more clear answer and more clean of a system also.
Good. Two more so far coming in. Lisa asked a follow-up question. When we get to applying this therapy to human patients, how long would it take from when the iPSC is generated to implantation in the patient's eyes?
So from iPSC to patient eyes, it's 10 weeks. But from the moment you collect the blood and you make the art, 10 weeks. In addition, you do all the assays, you wait for some reports. So yeah, I'll keep it 12 weeks or 12 to 15 weeks. Yeah, the most time consuming part that is iPS generation from blood cells, because that takes almost 2 months, and then you wait for the reports to come back. You want to make sure the iPS you make, they are real iPS. They don't have any, any kind of abnormalities that might affect the patient later on. So but yeah,
good.
It's a long process.
Eric asked a question again. Are you using VEGF promoters for the transplant of the scaffold, or has the newly transplanted RPE cells giving off enough VEGF to be reestablished to dilate the blood vessels to sustain cell life?
So so far we don't give the VEGF. We don't do the VEGF treatment when we implant the RPE. In our in vitro assays, what we have seen, and data I did not show, these RPE cells does secrete VEGF more on basal side, just kind of mimicking the in vivo part. So we believe that it is actually coming from RP cells that they are supporting. One of the reasons they might be supporting they are able to support the choriocapillaries is because they are secreting VEGF.
I think that's it. Well, no, here we go. Here's some more. Whoops, 1 second, guys. The question always comes in, and it's hard, and I understand your position, is and there's no magic, right? There's no magic globe or that you can see. When do you anticipate this happening potentially to get your trial to expand into phase 2, phase 3? And would it include choroideremia?
So yes, we are. I think if you we are working with different companies. We are trying to because clearly academia cannot do it alone. You have to collaboration with companies to do the phase 2, but the maximum we can do is phase 1. So we are in process of collaboration and actually transferring our technology to the company so that they can take to the phase 2. And what else sideways what we are doing is we are so far we haven't started working on choroideremia patient cohort, but what we are trying to do is there is another macular degeneration that choroideremia starts early in the age. Is MMD, and what we are trying to do, we are trying to start this trial with collaboration with some other company and labs, so that I think the idea is that we want to, because AMD is a late stage and the late age degeneration, we want to target the disease that happens early in the age. So I think hoping that MMD trial works and is success, I think that will be, it will be easily, that can help us in coming to the choroidemia group also. Yeah.
That's good. Here we have another question. I don't always understand the technicalities of research, but as most families who are desperate to see the end of the rainbow, here we are. What can we do? Is it funding? Is it putting pressure on politicians to cut through the red tape? Simply put, how can we assist you, wonderful people, such as yourself, expedite the end game? Excellent question.
Yeah, I think the little I know from my experience working in this field, I do think funding, because this is expensive. This is very expensive research for now. But I do think the funding is one of the things, but I do also think that doing several targeting the same disease in different ways will be beneficial too. For example, I did not talk about the drugs that we are using we are doing a phase 1 trial for macular degeneration and Stargardt. I'm not sure, I don't know if it's going to help the choroidemia, but I think those kind of things. So the way we are targeting, the way we are thinking is like stem cell alone won't help it. Drug alone won't help it in some cases. We are working to find drug molecules, we are working to find stem cells. And I think other thing that we need to do is like more of a partnership with pharma. Because I really don't think that, I mean, we do work with, we do collaborate with pharma, and we are tech transferring our technology so that they can take it to next level. But I think the collaboration needs to be done, and the funding gets in the way. But I think, it has to be more of a collaborative effort than one person.
I think that's it. I'm just looking that was a truthful answer. And one of the things that we continuously are doing at the choroideremia research, we are working with a very good ICRN. We have an SAB. We're funding a lot of research, and we have these types of webinars just to continue to provide that. The question behind that is hope and encouragement, and we're always on the watch, right? I mean, it's like TikTok, time is time. And we all are working that. Another comment, great talk. Thanks so very much. And I think Eric will be seeing you, maybe me, at coming up at the end of the month for Rare Disease Week.
Oh, nice.
So we'll ask some more questions. This is wonderful information, and
thanks.
We're grateful, super grateful.
Oh, I'm honestly, like, I feel like, thanks for having me. I think I would like to share what we know, and, in any other way we can help.
Mm-hmm.
Thank you.
The dial is moving forward, everybody. It really is. And so some of the people that you mentioned in your slides, David Gam, of course, he's working with us too. The best of the best is the goal. And that's what we're trying to do. And working at the NIH is important for us as well. So we thank you very much. Thanks everyone else. Hopefully we'll be getting more people coming on to share their work with us, and have a great Valentine's Day.
You too.
All right.
Bye,
Thanks,
everyone.
everyone. Have a good
Bye.
day. Bye-bye.